In this section, we discuss GLP as required by 21 CFR 58, EPA 40 CFR 806 and OECD. Now, what is special about this particular regulation? This regulation applies to a very special set of laboratories. Not all, not universal, but very special. To be liable under 21 CFR 58 or the OECD GLP, the nature of work undertaken in a particular laboratory should meet two important conditions, and both of these conditions. Now, what are they? The first condition is the non-clinical safety testing of test items contained in pharmaceutical products, pesticide products, cosmetic products, veterinary drugs, as well as food additives, feed additives, and industrial chemicals. These test items are frequently synthetic chemicals, but may be of natural or biological origin, and in some circumstances may be living organisms. The purpose of testing these test items is to obtain data on their properties and or their safety with respect to human health and or the environment. Now, the second and equally important condition, the data so obtained is intended for submission to appropriate regulatory authorities for licensing, registration, or approval for marketing. So what do we see here? This regulation applies to those laboratories and only to those laboratories which carry out non-clinical safety and toxicity studies. Any other study, this regulation doesn't apply. Now, the second and equally important condition is the purpose of collecting this data, analyzing this data, and submitting this data to a regulatory authority for the specific purpose of obtaining an approval, registration, or license. So even if the nature of the study is non-clinical safety and toxicity, but it is for purely academic purposes, the data generated is not intended for submission for any license or approval from any agency, then that laboratory does not come liable under this regulation. So these are the two important things. The nature of the study should be solely related to safety and toxicity, to man, animal, or environment. And B, the purpose of the study is to generate data for submission for approval or license to an appropriate authority in an appropriate territory. Now in GMP, the core values that the regulators seek to safeguard and promote are safety, efficacy, quality. Now in GLP, what is it that they are trying to safeguard? Let us see. The objectives of GLP: to assess the integrity of data reported by being able to have a third party seamlessly reconstruct the whole course of a study, even years after it has been performed, and even in the absence of persons having been actively involved in the conduct of this specific. The second objective is to place the responsibility and accountability clearly on individuals involved with the study. And lastly, to raise the awareness of good laboratory practices. Now, it could be a drug, it could be a cosmetic, it could be a food or feed item. So the US FDA gets involved. So you have 21 CFR 58. Now it could be pesticides which could harm the environment, and therefore the Environment Protection Agency has its own set of Code of Federal Rules 40 CFR 806. And the two combined is what comes under OECD, the Organization for Economic Cooperative Development. What is special about OECD is that it is an organization with member states and with mutual recognition in place. In other words, if a particular laboratory has been inspected and approved by one member country, that approval holds and is valid and recognized by all other member countries. Therefore, there is no replication of work, no duplication of work. Just as GMP has not found its final form overnight, so too GLP. There have been a chain of critical events that led to the culmination of these regulations. Now, what were these incidents? In the 40s, there was a sudden expansion in the number of chemicals that were being put into the market for various applications. But few people could assess the toxic effects these substances could have on man, animal, or environment. Around that time, there was a scientist considered one of the most knowledgeable in the subject, whose articles were read with a lot of attention and whose lectures were attended in large numbers wherever he went. This person was finally persuaded to set up a modest laboratory so that others who were not so well equipped or knowledgeable could give their substances to him for testing and assessing the safety and toxicity. So a laboratory called Industrial Biotest Laboratories was set up in 1953, and during its existence of 22 years, it carried out more than 22,000 studies relating to safety and toxicity. It soon became known for its reputation in terms of its high science and quality of its work, as well as the very moderate terms that were charged per study. With the success of one unit, there was a mushroom of a large number of similar units of varying scientific quality. Biometrics was another large laboratory. By the 60s, the Biotest Laboratory had become so famous that almost 2/3 of the licenses submitted worldwide had their safety, toxicity, material safety data generated and cleared by Industrial Biotest Laboratory. By the early 70s, if any regulator anywhere saw the Industrial Biotest stamp, he would just quietly move on and pass the application, approve the application. All this came to a grinding halt in the mid-70s with serious incidents reported in the field. And we had what is called the famous Kennedy hearings, going into the investigations of the kind of practices that were in place in these various safety and toxicity testing laboratories. These were all contract research organizations. Now, you don't run a contract research organization on the basis of a few dollars here or there. Each of the assignments runs into hundreds of thousands of dollars. Now, no sponsor is willing to put $100,000 on the table if at the end of 3 years or 2 years you go back and tell him that that material is not suitable for the intended use. The sponsor would always like to hear what he likes to hear, that the material is indeed safe. Now, if a contract research organization were to repeatedly say that some material is not acceptable, it is quite likely that that sponsor would go to some other laboratory who's willing to give more favorable results or interpretations. So the pressure was always there on the contract research organization to somehow or the other give the kind of results or reports that the sponsor would view with favor so that more business could come from him. So as a net result of all this, we had a situation where what the Kennedy hearings found out was a can of worms, the largest scientific fraud that had ever been committed till date. Animals that died were quietly replaced without documentation. Animals escaped and there was no documentation. In the raw data, there is a mention that the control animals had a benign tumor. But in the final report, these benign tumors were altered to say the control animals had malignant tumors so that it compared more favorably with the final results. In the case of flagell, for example, in the final report, it was mentioned that in a study relating to carcinogenicity, that there was a tumor, mammary tumor in a male. Now, mammary gland tumors are common in females, but not quite so in males. So when they asked for the raw data, somehow the raw data was not traceable. And finally, they asked for the specimens, and the answer came back saying that the tissues were no longer usable or testable. So the suspicion lingered that perhaps the male control animal in the raw data subsequently was replaced with a female animal without recording it. Autopsy reports were conveniently missing for 50% of the study or 30% of the study, and all sorts of such things had happened. So much so that the regulators felt that it was high time that some discipline was brought in place for the conduct of a study, for the evaluation of a study, for the assessment of a study, and the manner in which the findings were reported. Now, it's very heartening to find the structure of 21 CFR 58 and compare it with the structure of 21 CFR 210211. In the same pattern, the regulators have looked at the three aspects of GMP or GLP. The resource-related aspects, the operational or manufacturing aspects, and the controls-related aspects. So just as we have in 21 CFR 211, subpart B, personnel, subpart C, buildings and facilities, subpart D, equipment, so too you find in 21 CFR 58 the same pattern being followed. Subpart B, organization and personnel, testing facility management, study director, and the quality assurance unit. In subpart C is the facilities, the general requirements, animal care facilities, animal supply facilities, facilities for handling test and control articles, laboratory operation areas, and specimen and data storage facilities. And subpart D talks about equipment in terms of equipment design, equipment maintenance, and calibration. Now, subpart E, F, and G refers to the study itself. Testing facilities operations, subpart E, standard operating procedures, reagents and solutions, and animal care. And subpart F, test and control activities, test and control article characterization, test and control article handling, mixture of articles with carriers. And subpart G, the protocol for and conduct of a non-clinical laboratory study, the protocol itself, and the conduct of the non-clinical laboratory study. Subpart H and I, which in GMP relates to holding and distribution as well as laboratory controls, they've reserved that and gone on to subpart J, which is the records and reports as in GMP. Reporting of non-clinical laboratory study results, storage and retrieval of records and data, retention of records. Subpart K is the main difference between the two. Whereas in the case of GMP, it relates to salvage. Here it is a very important issue of disqualification of testing facilities that includes discreditation of the personnel associated with the study in the event of wrongdoing or outright fraud. The OECD GLP is organized slightly differently. The first part is about the introduction to the OECD principles of GLP, which contains the introduction, the fundamental points of GLP, and the OECD GLP principles. The second part refers to the resources in terms of personnel as well as facilities, that is buildings and equipment. The third section talks about the rules, the protocol or study plan and the SOPs. The fourth part talks about characterization of the test item as well as the test system. The fifth part refers to the documentation, the raw data and data collection, final report, the archives and the indexing. And the last part refers to quality assurance unit, the final report, the raw data audit, quality assurance statement, quality assurance inspections of suppliers and contractors, the distribution and archiving of quality assurance unit files and reports. Now there are what you can call the pillars that hold GLP in place. One leg or one pillar is the regulating authority, and the other is the management or the sponsor. The third is the person who actually plans and carries out the study, evaluates, assesses and submits a report. And the fourth pillar is the quality assurance. The quality assurance unit shall be reporting directly to the sponsor or to the management and will have no interaction directly or will never report to the study director or vice versa. The study director is independent and he reports to management of the sponsor. The quality assurance unit also reports directly to the sponsor or management. The purpose of the quality assurance unit is to audit the raw data, check whether it is in line with the study protocol and ensure the authenticity or the integrity of the data that is being collected. And finally, they issue a statement stating that the study was indeed carried out in full compliance with GLP requirements. Now, a test laboratory may be a very small unit, or could be a very large unit, or could be something in between. If it is a very small unit, they can't possibly afford to have their own quality assurance unit, so they are recommended to outsource it. If it is a large enough laboratory or facility, they are quite in a position to have, like any pharmaceutical drug manufacturer, an in-house quality assurance unit, full-fledged unit. But if you lie in between, then GLP regulators offer you a very special concession. Now let's see what that is. If you have a test facility management and there are two test facilities under two different study directors, study director 1 and study director 2. Under study director 1, you have unit A, unit B and unit C with analysts M, N in A, O, P, Q in B and R in C. In test facility 2, under study director 2, you have the units X and Y, where the analysts are S, T and U in X and V and W in Y. Now let us say study 1 has activities in A, B and C. Then it has nothing to do with unit 2 or test facility 2. QA could be carried out by the analyst, anyone from S to W, and similarly the audit of test facility 2 could be done by any analyst, M to R. In the case of study 2, where there are activities in units A, C and X, then QA could be carried out by analysts O, P or Q for the part in unit X and by analysts V and W for the part in units A and C. Now what does this mean? It means the important requirement for quality assurance is that it is independent of the study. The person who's carrying out the audit should not be involved in any way, directly or indirectly, now or subsequently, with that particular study. Should be totally independent. Now, should that person be a subject matter expert? No. The study director is the subject matter expert. But The analyst should have enough knowledge to be able to intelligently carry out with due diligence the audit assignment given to him. For example, if he is checking out gas chromatographic work, he should be a specialist in gas chromatography. He need not be a specialist in the kind of study that is being carried out, but as far as his area of audit is concerned, he should be a specialist. But he should be independent of that study. So, he is working as a person, as a regular analyst in one particular unit, and as a part-time effort, he doubles up as a QA for audit in another unit in which there is a study going on with which he is not in any way directly or indirectly involved. This is a very special concession that has been given in the GLP environment for organizations that are modest in size, that cannot afford a full-fledged quality assurance unit, and for whatever reason do not have the option of outsourcing a specialist for carrying out the audits. Now, as in GMP, there are always tricky situations. What if the quality assurance person, not involved with the study in any way, but is in some way related to the study director, his wife or her husband or son or son-in-law or whatever, is there conflict of interest? So these sort of issues do come up in GLP, where you have to establish not only is the person carrying out the quality assurance activities independent of that study, but also has no conflict of interest with the personnel or the study that is being carried out. The quality assurance person is also a member of the top management. Then where is the question of independence? So these are all technical issues that do come up from time to time in GLP as they do in GMP. Now, GLP governing laboratories carrying out safety and toxicity studies is a subject by itself as big as GMP. And in the QC laboratory or QC environment in the pharmaceutical industry, all those don't all apply. But nevertheless, it helps to have a basic idea of what is it that those regulators are focusing on. So the first thing that comes up is the test facility or test facilities. The next is the test site or test sites, maybe more than one site. Then comes the nature of the study. Is it a long-term study or a short-term study? Now, what is the difference? In the long-term study, retention of samples of test items is compulsory. Every study plan should be unique and the study reports duly reviewed and approved. The study plan is a study protocol that has to be pre-approved before the work can commence. And finally, there is a QA inspection of the study from start to finish. In contrast, we have the short-term study. Now, short-term and long-term doesn't necessarily have to do with the time it takes. For example, application of a pesticide may take half a day, but the results will come in over a period of 3, 4 or 5 years. Now, what is it, a short-term or a long-term? So, the short-term study specifically relates to that kind of activity which is routine in that laboratory, which is commonly carried out in that laboratory, regularly carried out in that laboratory. Melting point, boiling point. If a laboratory carries out this study once in 10 years, then it is not routinely carried out in that particular laboratory. So it is as part of the long-term study. It's a special study. But if this is done 10 times a day by each analyst, then it is a routine study. So in the case of short-term studies, the regulators have made certain concessions. And what are they? First, you need not retain the test item. B, you can have a template that is pre-approved that could be routinely used. You don't have to prepare and get approval for a study each time uniquely. One common template, pre-approved, could serve for all such tests. And 3 is quality assurance doesn't have to audit every study. It could do it on a periodic basis, once in a quarter, and statistically pick that many records and audit them. They don't have to do 100% of auditing. Then you have what is called the test item, which is the substance whose safety and toxicity that you are trying to assess. And the test systems. Test systems could be an animal model, a plant model, and marine model, whatever. That test system should be established. It should be fully characterized and it should be established as appropriate for this kind of study. And then you have the study plan or the protocol, which gives full information as to how you plan to do your work, how you plan to proceed, what results you expect, and how you propose to gather the raw data, analyze the raw data, evaluate the raw data, and make an assessment. And then you must have deviations and change control systems in place for amendments. Then the raw data as to how it will be collected, how it will be handled, how it will be stored. The phases of the study, it could be in one phase, it could be in multiple phases. And then you have what is called the master schedule, from start to finish at one site or several sites, at one location or several locations, under one principal investigator or several such principal investigators with research scientists working under them. When do you propose to start and when do you propose to complete? The dates to be involved. The study initiation, the study start and completion dates, the study performance and reporting, the auditing schedule, and finally the archives. Here we see the highlights of GLP as a quality system. First, you have an approved study plan or protocol. There should be a unique identification for the study and all of its parts with change control. All original observations in a study should be immediately, clearly, and legibly recorded. The recording should be indelible and corrections should be made so as not to obscure the original entry. For all corrections, the respective reasons have to be provided. All entries and corrections to them should be dated and initialed. All records should be in the form of bound notebooks or on continuously numbered sheets. Records related to the test system itself for biological test systems, the acquisition, condition, suitability testing, etc. For physical chemical test systems, their specifications, manufacturer, model, etc. These should be gathered and retained. Specimens should be clearly identified so as to allow full traceability. At the end of a study, all raw data should be assembled, catalogued, and archived. Archiving should provide for secure storage and retrieval of all raw data, samples, and specimens, together with any other documents such as study plan and study report, if any has been written. Now, why GLP principles are worth embracing even in non-GLP environments? What do we mean by non-GLP environments? We refer in the specific context of 21 CFR 58 or 40 CFR 806 EPA. We are referring to those environments where we do not carry out studies relating to safety or toxicity, but something else. So even in those environments, how these GLP principles are worth embracing. GLP is a quality system particularly developed for laboratorial work where data integrity is never compromised and full accountability is maintained. In an environment where publish or perish pressures prevail, GLP protects against fraud while at the same time protects those wrongly accused of scientific misconduct. Now, what does this really mean? You see, if you are in an academic institution, or for that matter any institution, for your research you need funds, and there are limited funds, and funds are disbursed based on the kind of work you have been doing in the past, your track record. For example, a scientist who has more papers published in prestigious journals stands a better chance of funds than another scientist whose reputation of track record is not so eminent. So publish or perish. Now here is a situation where there was a lady, very highly respected, very good scientist, who was reaching the end of a certain study on behalf of a certain sponsor, who was the market leader in that particular field and was moving into the next generation of product. Now he already had 70% of the market, and if this study was approved, then he could release the new item in its new avatar, and then he is more or less going to wipe the market off. So his competitors got very, very nervous. One of those competitors wrote an anonymous letter to the regulators saying whatever work this lady had done was pure basic fraud. This triggered an investigation, and that lady had used all loose sheets and pencils, and when she collected the raw data, she just rounded it off instead of having the original raw data present. It took 10 years for the regulators to assess her work and declare her innocent. But in the process, the competitors had achieved their objective. They wanted to buy time so that they too could come out with some new product. And by the time this new item could be released in the market, the competitors had already protected their turf. So here was a case, had she been more careful in the way she collected her raw data and handled her practices, she might have been able to vindicate herself much earlier. There was another case of another lady who had done some considerable work in a particular field and then had gone on holiday. When she came back, she was horrified to find that one of her colleagues had actually published a paper, and the data that was mentioned appeared to be very much from her own work. So she cried foul and called the regulators, who went across to the other person and found that he really had no raw data. All he had really done, he confessed in 3 months, was that he had cut paste from this lady's work and published it as if it was his own. So GLP helps those who are innocent to protect their innocence and prove their innocence, and GLP protects against scientific fraud or misconduct by those wanting to abuse the system. The study plan protocol is often accused of stifling flexibility in imaginative approaches to and science-based conduct of research or study. But the flip side of that, it helps one marshal one's thoughts and plan ahead meticulously. Change control protocols in place do permit deviations or alternatives. Now, what does this mean? Most scientists violently protest against imposition of GLP, especially that so-called submission of the study plan or protocol. Their argument is that we are research scientists. We are on a voyage of discovery into the unknown. If we already knew what are the results that we are going to come up with, then where is the question of research? So it is very difficult for us if you stifle the freedom that we have to think out of the box and come out with science-based approaches to solving problems, because as a research scientist, I do not know what I shall be doing this afternoon. What I shall be doing this afternoon will be determined by the direction in which the data of the work done in the morning is pointing. If it is pointing in a certain direction, I pursue that in the afternoon, and depending on what direction the data of the work done in the afternoon is pointing, that is where I will resume my work tomorrow morning. As of now, I have no clue as to what I shall be doing in the afternoon or tomorrow morning, leave alone what I shall be doing over the next 6 months or 6 years. But on the flip side of it is, if you were to sit down and actually marshal your thoughts and write down, how is it that you want to do? You always have change control in place. You're permitted to make deviations, but through change control. If you have a road map, then chances are that you will be able to recognize your destination when you get there. If you move without any kind of a roadmap, you're very unlikely to be able to see that you've done your work in the most efficient and most scientific way possible. That is the argument coming from the other side. The requirements of SOPs for all routine and non-routine activities with change control protocols, maintenance, calibration and calibration documentation for apparatus, instruments and equipment. GLP instills discipline with regard to raw data management, timely, accurate documentation and recording habits. Procedure for raw data handling, recording and reporting. So here we see how GLP principles may be applied in non-GLP environments. There should be an approved protocol with a unique identification covering all of its parts with change control. All original observations should be immediately, clearly and legibly recorded. The recording should be indelible and corrections should be made so as not to obscure the original entry. For all corrections, the respective reasons have to be provided. All records should be in the form of bound notebooks or on continuously numbered sheets. All entries and corrections to them should be dated and initialed. Records related to the endeavor itself should be gathered and retained. Specimens should be clearly identified so as to allow full traceability. At the end of the exercise, all raw data should be assembled, catalogued and archived. And archiving should provide for secure storage and retrieval of all raw data, samples and specimens together with any other documents and reports. So what do we really see here in this GLP as a quality system? It has two elements. One is the good science part of it and the other is the good organization part of it. In the good science, you have careful definition of experimental design and parameters, science based on known scientific procedures, control and documentation of experimental and environmental variables, careful, complete evaluation and reporting of results. Results become part of accepted scientific knowledge. In the organization part, provision of adequate physical facilities and qualified staff, planning of studies and allocation of resources, definition of staff responsibilities and training of staff, good record keeping and organized archives, implementation of a process for the verification of results and compliance with GLP. GLP is a quality system concerned with the organizational process and the conditions under which non-clinical health and environmental safety studies are planned, performed, monitored, recorded, archived and reported. It is not a system that guarantees the scientific validity of the method used in any study or the ability of the test facility to generate accurate and precision measurements or the instrument that assures high scientific standards. So what does this really mean? It means if there are two test facilities and both of them have been accredited, both of them have been approved as being GLP compliant, it does not automatically guarantee that the quality of the work done in one facility is of the same order as the quality of the work done in another facility. If one company is ISO 9000 certified and another company is ISO 9000 certified and both make the same products, the fact that they are ISO certified does not automatically imply that the products that they produce are of the same high quality. All it really says is, here is a test facility with a system in place that permits seamless back audit trail. And here is another test facility which also has a system in place, quite a different system in place perhaps, but a system nevertheless that permits seamless back audit of all its activities. The trail is complete. That is what we want in GLP as per 21 CFR 58. A seamless reproduction, reconstruction of all activities reported to arrive at the same results that were reported by having people carrying it out in some other place, in some other facility without reference to the people who were originally associated with the activity and even years after it has been carried out, even in the absence of the persons who had originally been part of the study.